Hello, welcome to OK Biology Classroom and welcome to our lesson 4.3.2 on DNA replication. Remember that in 4.3.1 we talked about the same process but in a simplified form. In this lesson we are going to look at the same process but in an expanded form. But before we get into what we have for this particular lesson, please remember to subscribe to our YouTube channel so that you always have our videos whenever they are released. DNA replication. At the end of this lesson, the student would be able to describe the roles of enzymes and primers in DNA replication. In 4.3.1, we did not talk about the roles of the specific enzymes involved in the various stages of DNA replication, but that is going to be included in this lesson. As well, we did not talk about the roles of primers, so we will be talking about roles of primers in this lesson. We are going to use the same stages as we did in 4.3.1. So we talked about DNA replication in 3 stages. The stage 1 being the unwinding and unzipping of the original DNA molecule. Then stage 2 is when the new strands of DNA are formed against the original strands. And finally, there will be zipping and winding to form 2 new molecules of DNA. Right. So we are going to use the same stages as I said. So let's start with the stage 1, unwinding and unzipping. In lesson 4.3.1, I talked about the unzipping of the DNA molecule, that is the original DNA molecule. And the unzipping would mean that the hydrogen bonds that hold the nitrogenous bases are broken, so that they would be separated. And note that the separation is not as simple as I've presented it here, because the DNA molecule is actually a very long one. So because of space, I have only concentrated on part of it to show to you how the unzipping occurs. But actually, there is a long molecule of DNA, which the unzipping process would also continue on that particular part as well. The only information that I would like to add in this case is the enzyme involved in the unzipping process. The enzyme is called helicase. So here I'm saying that during unzipping, an enzyme called helicase breaks the hydrogen bonds between the nitrogenous bases, and that is what leads to the formation of the 2 strands of DNA. It starts from one end of the DNA molecule to the other end. Right. So this is the information that I have for you for the stage 1. It is the same description as in 4.3.1, but what I'm saying and adding in this particular case is that the enzyme involved is called helicase for the unzipping of the DNA molecule into the separate strands. So now let's move on to stage 2. Stage 2 is about formation of the new strands. Let's go back to what we talked about in 4.3.1. Yes, I said that as the 2 strands of the DNA molecules are separating, new nucleotides, that are the free nucleotides of DNA within the nucleus, would assemble themselves against each of the strands. So against one of the strands, in this case I referred to as the upper strand, new nucleotides would assemble, and against the lower strand as well, new nucleotides would assemble. But the point is that the process is not as simple as it was shown in our lesson video 4.3.1. So here we are going to look at it, exactly what happens. So let's look at the details of stage 2. Let's look at this particular image, which is showing portion of a DNA molecule, because I've said that the DNA molecule is so long, so I cannot show the whole DNA molecule on the screen here. So we are only considering part of it. And even in this case, we're going to consider just a portion of this particular DNA molecule. Right. So you can see clearly here that there is unwinding and unzipping of the DNA molecule. And this is going to occur, as we have already been made aware, that the unzipping is going to occur along the entire length of the DNA molecule. So here, I will concentrate on the part that I have highlighted in this case. So when that is expanded, we are going to see something like this. So you're going to see 2 strands, which in the first lesson video, I said, or I referred to them as the upper strand and the lower strand, right? Now let's look at something about these 2 strands, which I'm going to use to do the explanation. Every DNA molecule has two ends. So we are going to consider them strand by strand. So let us consider the strand at the top in this particular case. So what we can call the upper strand on my screen here. This strand has a 3' end and a 5' end. In fact, for our level, I'm not going to explain into detail what makes us refer to one end as a 3' end and what makes us refer to the other end as a 5' end. But what we need to notice that each strand has a 3' end and a 5' end. So let us look at it. Let's consider the upper strand first. There is a 3' end here. And since one side of it or one end of it is 3', the other end of it would be 5'. It is as simple as that. Let's look at the lower strand. Considering the right hand side of the screen, you would see that the upper strand has a 3' end there. Whenever one strand has a 3' end, the complementary strand would have a 5' end. So looking at this lower strand having a 5' end at its right hand side of our screen, it means the left hand side is going to have a 3' end. So here you can see that they are reading differently. If you consider reading from our right hand side to our left hand side of the screen, what you will see is that the upper strand would be reading from 3' end to 5' end. But the lower strand is going to read from 5' end to 3' end. In this case, the strand which is reading 3' end to 5' end on our screen, that is the upper strand in this case, we are going to refer to it as the leading strand. And the strand which is reading 5' end to 3' end, we are going to refer to as the lagging strand. So now let's bear that in mind, that there is a lagging strand and there is a leading strand. And we're going to use these terms a lot in our subsequent explanations. After identifying the leading strand and lagging strand in this case, let us now look at the second stage into detail, how the new strands are formed against the original strands, because what I'm showing here are the original strands. So let us look at how the new strands are formed. Now I have separated the leading strand and lagging strand enough. So let us consider the leading strand first. Right. In lesson 4.3.1, I talked about the arrangement of free nucleotides against the original strand. But the process is not as straightforward as I presented it in that particular lesson video. Well, I wanted to use that as an opportunity for you to get a summary of how the process actually takes place. But here you need to get the additional information which would complement your knowledge on the process of DNA replication. The first point I would like you to notice that there is an RNA called primer. The primer is what starts the whole process, and note also that this primer is produced by an enzyme called primase. Let us consider the leading strand. What would happen next is that this primer would be attached to the 3' end of the leading strand. So like this. Now, after the primer has been attached to that end of the leading strand, the next action is that an enzyme called DNA polymerase would now be attached to the end of the primer. So like this. After the DNA polymerase has been attached to the end of the primer, this DNA polymerase, which is an enzyme, is now going to continue the arrangement of the free nucleotides of DNA along the leading strand. So in this manner, from the 5' end to the 3' end, in this direction. So like this, free nucleotides are arranged by the DNA polymerase against the original leading strand. And this happens along the entire length. Remember that I told you that because of space, I am only limiting myself to a part of the DNA molecule. So they will be arranged along the entire DNA strand so that a full complementary DNA strand would be formed. But note that in this case, the strand still has a portion of it being a primer. So there is a primer that is attached at the 3' end of the original leading strand, but the rest of it has been occupied by free nucleotides of DNA. So there are DNA free nucleotides and then there is a primer, but the strand is now full. Let us now move to look at what would happen on the lagging strand. In the case of the lagging strand, the arrangement wouldn't be like that of the leading strand. Well, of course, there would be some similarity which I'll be talking about very soon. What we need to notice that In the case of the lagging strand, the primers are inserted at intervals. So they are not inserted at the 3' end of the original strand, as it happened in the case of the leading strand. You remember, in the case of the leading strand, the primer was inserted against the 3' end of the leading strand. But in this case, it is not going to be so. The primers are going to be inserted at intervals, so that the spaces that are left would now be filled by the free nucleotides of DNA. So let's look at a situation like this. You can see a primer inserted here, right? And after the primer has been inserted, the next action is that DNA polymerase would be attached to one end of the primer, so that it would continue with the arrangement of the free DNA nucleotides from the 5' end to the 3' end direction, the same as it happened in the case of the leading strand. So you can see that one similarity in the processes involved in arrangement of free nucleotides against the leading strand and that of the arrangement against the lagging strand. The similarity is that the arrangement is always from the 5' end to the 3' end of the new strand which is going to be formed, not of the original strand. So note that the direction we are considering here is the direction of the new strand which is being formed. So from the 5' end to the 3' end. So you can see it going in this direction, 5' to 3'. So you can see that it will be attached, or the DNA nucleotides are going to be attached from here, because that will be the direction. So like this, until completed. Now, because they are going to be inserted at intervals, the process will be repeated at the other parts of the lagging strand. It means that primers will be inserted, and as primers are inserted, the nucleotides are also inserted accordingly. So next primer would be inserted this way, and the free DNA nucleotides would be inserted after the DNA polymerase has been attached. So in this manner. Right, so it tells us that the difference that we see in the case of arrangement of DNA nucleotides in the case of the leading strand and the case of the lagging strand is that in the leading strand, what happens is the primer is inserted at the 3' end first. Then the DNA nucleotides are arranged along the entire length of the leading strand. But in the case of the lagging strand, the primers are inserted at intervals, so not specifically at the beginning of the molecule. Right, but at intervals. Okay. So now we can see that both the leading strand and the lagging strand have free nucleotides inserted or paired against each of them. So new strands have been formed. But the only problem is that the new strands which have been formed are made up of primers as well as DNA nucleotides. So the next action is that these primers would have to be removed. The primers are removed by an enzyme called exonuclease. So after exonuclease has removed all the primers, then DNA nucleotides are inserted by the DNA polymerase. So you can see here that all the primers have been removed. So let's look at, after primers have been removed, how the DNA nucleotides are inserted on the leading strand. So like this, so that they will fill the space that was occupied by the primer. The same thing would happen on the lagging strand. So the spaces which have been left would now be filled by the free DNA nucleotides, and the work is done by the DNA polymerase. Right. And this side too would also be filled in the same direction, 5' to 3' end, and is sealed. So after this has been done, and remember that it will happen along the entire length of the DNA molecule. And I repeat, this is not the full DNA molecule. I told you that I'm only focusing on a length of the DNA molecule as far as my screen would allow me. Right. So it would continue on the other parts of the DNA molecule until the entire length of the DNA molecule has new strand formed against each of the leading and lagging strands. Well, we may say that this means the whole process is complete, but not yet. What needs to be done is proofreading to finalize everything under the formation of the new strands. So that is in our stage 2. So the final stage of our stage 2 would be proofreading. And what does it mean to proofread? You know that even in our writings, what we do is that sometimes you write something and you would have to read over again to see that there is no error in what you have written. And a similar thing happens in here. The arrangement of the free DNA nucleotides may have had some errors in them along the line. So there is proofreading. So DNA polymerases would now check if there are errors in the pairing and make corrections. So if there were problems with how some of the nucleotides were paired, then these DNA polymerases would correct them. You know the pairing rule. Adenine is supposed to pair with thymine. Cytosine is supposed to pair with guanine. That is how the pairing should go. So they would have to check to see that all the pairings have been done correctly before finalizing the whole process. After the proofreading, then we would say that the stage 2 is now completed. So we can move to the final stage, which is stage 3. And with the stage 3, it's about zipping and winding, just like we talked about in our previous lesson. What happens in this case is that the nitrogenous bases of both strands would be reconnected by hydrogen bonds, because remember that in the first stage, hydrogen bonds were broken to unzip the original DNA molecule. But now we have one original DNA strand and a new DNA strand formed, so they would need to be reconnected. So the reconnection would mean that hydrogen bonds would have to be reformed, and the enzyme responsible for this is called ligase. Right. After ligase has done its work, that is going to mean that there would be winding again into the helical structure, so that there will be formation of two new DNA molecules. So like this, as you're going to have each of them forming these two new DNA molecules. Remember that this one is made up of one old and one new DNA molecule, and this would also be made up of one old and one new DNA molecule. I said this in lesson 4.3.1 that DNA replication is very important because new cells are going to be formed, and each of these new cells must have the same DNA information as in the old cell or in the original cell. Right. So when the process occurs this way, it gives each of them the exact information as existed in the original DNA molecule. So that is the whole process of DNA replication for now and for our stage. This information would be enough for us to understand how the DNA replication process occurs. But before I end, let me touch on why we refer to the process of DNA replication as semi-conservative. Well, DNA replication is called semi-conservative because the new DNA molecule is always made up of one old and one new strand. There is always an original strand and a new strand. So since the original strand is always part of the DNA molecule, that is what we base on to refer to it as semi-conservative. Right. So that brings us to the end of our lesson for today. So I have said that DNA replication process could be summarized in three main stages. So I talked about unwinding and unzipping of DNA molecule. Then I came to talk about formation of new strands against the old strands. And then I talked about winding and zipping of two new helical DNA molecules. Right. In our next lesson, we are going to look at protein synthesis. But before we get into that, I have a few questions for us to try. The first question is that state the role of each of the following enzymes in DNA replication. So the first of them is DNA polymerase. The second one is helicase, then primase. Then also, what does the semi-conservative process of DNA replication mean? That is my second question for you. And finally, state one function of the primer. Right. I will provide an email address as well as a WhatsApp number so that you can send your answers to them. Okay. And if you have any questions too, you can send them to my WhatsApp number and I'll respond to them. Okay. But before we end, remember to subscribe to my YouTube channel so that you always have the videos whenever they are released. Thank you for being part of today's class. See you in our next lesson on protein synthesis.